last session. Students, the last session until where we had discussed.
So digestion in stomach.
Stomach, sir.
Digestion in stomach only. Stomach. Digestion in stomach only. Food not received in intestine still?
No, sir.
Not received.
No, sir.
Very good. Right. Okay, students. So, in earlier session, we had discussed that the process of the digestion takes place in the region of the stomach. So, in the stomach, which digestion takes place, students? Which material digestion?
Protein.
Protein.
Protein digestion is initiated or completed in the stomach?
Initiated.
Initiated, right. So, in the stomach, after the churning process, what is formed? The food is called?
Chyme.
Chyme, right. So, next one, the digestion process in small intestine. Yes, students, from the stomach, the chyme enters from where to where? Stomach to
Duodenum of small intestine.
very good. Duodenum part of small intestine. So in the region of duodenum, the process of digestion takes place. Final digestion takes place by the help of secretion of liver, secretions of pancreas, and secretions duodenum itself. Clear, students? So where the final digestion process occurs? In the part of duodenum. Why in the part of duodenum only final digestion takes place? Because of the duodenum receives all the digestive secretions from liver, pancreas, and whatever the secretions released from the duodenum. Clear, students? That point you need to remember. So, the duodenum receives all the secretions of what, students? Secretions of liver, secretions of pancreas, and digestive glands of duodenum also. Digestive glands of duodenum. So that only the final digestion process. So whatever we'll discuss now, all the enzymatic reactions which takes place in the region of duodenum only. The final digestion process takes place in the region of duodenum. Clear, students? So in duodenum, final digestion process takes place.
Sir, the board is not clear, sir.
Takes place. What is not clear, students, for all?
Clear, sir.
Clear, sir.
Okay. Check your net connection, man. Internet connection. Right, students. So where the final digestion occurs, students? In which part of the intestine?
Duodenum.
Part of it.
Duodenum, the final digestion process takes place. What is the reason, students? Because of
It receives secretion from liver.
all the secretions from the liver, pancreas, and duodenum itself has some of the digestive glands. Okay, students? Right. So, in the duodenum, so what type of enzymes are secreted? and what type of the materials are presented, which enzymes are secreted from the region of duodenum. So duodenum, the submucosa has, the submucosal layer has which glands? Specialized glands are presented. Those are called Burners glands. Burners glands. So what is the location of Burners glands in the duodenum? Burners glands are formed from which layer of the part of the gut? That is submucosa. From the submucosa, modifications are Burners glands, and Burners glands secretions. Burners glands secretions helps to maintain alkalinity in duodenum. In the cavity of the duodenum, always alkalinity maintenance is required. Why, students? Because of all the enzymes works in the cavity of the duodenum, in the pH level, alkalinity only. In the acidic nature, it cannot, the enzymes cannot work properly. Okay, students? So in the cavity of the duodenum, the alkalinity is maintained by the presence of secretions of Burners glands. Where the Burners glands are presented, which layer of duodenum, students? That is most important. Which layer of duodenum? Submucosal. That point is most important. Okay, students. And along with that, mucus plus bicarbonates also. The mucus and bicarbonates also, both are also helps to maintain, yes, students, help to maintain alkalinity in duodenum. So by these 2 points, what you came to know, all the enzymatic reactions which are takes place in the region of the duodenum, in which pH, students? Acid or base?
Base.
Base.
Alkalinity is maintained. So how alkalinity is maintained constantly in the region of cavity of the intestine duodenum? Because of secretions from the Burners gland and mucus, and there is bicarbonates. Mucus, bicarbonates, and secretions of Burners glands helps to maintain alkalinity in the region of cavity of intestine, cavity of the duodenum. Okay, students, right? Now, in the region of the duodenum receives bile juice from, yes, students,
Liver.
bile from
Liver.
pancreas, juices from
Pancreas.
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 Through which duct, students?
A part of pancreatic duct.
So before discussion of enzymatic reactions, which enzymes are secreted from pancreas, and what is the components? What are the components are presented in the bile juice? We came to know. After that only we will go to discuss, and after that only enzymatic reactions we are going to be discussed. Okay, students. So first one, the enzymes secrete from the pancreas. The list of enzymes you need to remember, very most important, list of enzymes and function of the enzymes. Clear, students? After that, we will discuss enzymatic reactions. So from the pancreas, what type of the enzymes are releases? The enzymes releases from the pancreas, trypsinogen, pancreatic enzyme, right now. Pancreatic enzymes, trypsinogen. Second one, chymotrypsinogen, Third one, procarboxy peptidase. Fourth one, nucleases. In the nucleases two, ribonuclease. Next one, deoxyribonuclease. Okay, next one, pancreatic amylase. Next one, pancreatic lipase. So these are the enzymes secreted from the pancreas. Pancreatic enzymes secreted, the enzymes secreted from the pancreas, pancreatic enzymes released into, yes students, released into where?
Duodenum.
Released
Duodenum.
into duodenum. Otherwise, you can write duodenum receives below following pancreatic enzymes. So the pancreatic juice consists with the different types of the enzymes. First, you need to know what are the enzymes released from the pancreas. Those are trypsinogen, chymotrypsinogen, procarboxypeptidase, nucleases. Those are ribonuclease, deoxyribonuclease. Next one, pancreatic amylase and pancreatic lipase. Clear students, these are the list of the enzymes. Now, what are the functions of these enzymes? Trypsinogen and chymotrypsinogen and procarboxypeptidase all are proteolytic enzymes. Proteolytic enzymes. Proteolytic enzymes means influences what we speak, students? Very good. Proteolytic enzymes.
Protein enzyme.
Proteins act as a catalyst on proteins. Next one, nucleases in the name only.
Nucleic
Ribonuclease,
acid.
deoxyribonuclease, ribonuclease and RNA, deoxyribonuclease on DNA, ribonuclease and deoxyribonuclease. Next one, pancreatic amylase. Amylase always works on
Carbohydrate.
Carbohydrates. Starch.
for carbohydrates and the backbone of carbohydrates. Next students, lipase. Lipase always acts on
Lipids.
fats,
Fats.
lipids, fats and lipids or lipids.
Yes, sir.
Okay.
The board isn't clear, sir.
What is not clear? Paral?
Clear, sir.
So it's clear.
Okay, right, fine. So these are the enzymes separated from the pancreas. And enzymes, what are the functions of the enzymes? Okay, students. Now, this points again I'm revising. So the duodenum receives all the secretions of liver, pancreas, and digestive glands of duodenum. Yes, students. So in the region of duodenum only, why the final digestion occurs? The reason is because of all the digestive enzymes received by the duodenum part of the small intestine. Next one, duodenum submucosal has, the submucosal layer has Brunner's glands, the secretions of the Brunner's, which helps to maintain alkalinity in duodenum. All the enzymes works in alkaline condition only, means in the condition of base condition only. So mucus plus bicarbonates, mucus and bicarbonates also helps to maintain alkalinity in the region of the duodenum. Clear, students? Next one, the pancreas, the duodenum receives all the enzymes which is secreted from the pancreas. So pancreatic enzymes, what are the pancreatic enzymes? Pancreatic enzymes are trypsinogen, chymotrypsinogen, procarboxypeptidase, ribonuclease, deoxyribonuclease, pancreatic amylase, pancreatic lipase. So trypsinogen, chymotrypsinogen, procarboxypeptidase acts on what, students? Those are proteins.
Proteins.
Acts on proteins. And ribonuclease and deoxyribonuclease acts on DNA and RNA, nucleic acids. Okay, students, you can write the nucleic acids in the bracket. Right, students? And amylase. Amylase and what, students?
Carbohydrates.
Pancreatic amylase and what? Carbohydrates. Pancreatic lipase and what, students? Fats and lipids. Until this clear, all of you? Yes, students?
Substance is secreted by
Yes, sir.
The
Yes, sir.
digestion
Clear, sir.
enzymes which are secreted from the pancreas we discussed. So, the different types of the food materials digestion, which is influenced by pancreatic enzymes. The food materials are different types of the food materials influenced by the pancreatic enzymes. And what type of the reactions are takes place now we discuss. Okay, students. So these are all enzymes releases from the pancreas and acts in where?
Duodenum.
Acts in duodenum.
Duodenum.
That point you need to remember. Clear, students? Releases from the pancreas and acts in the region of duodenum. So the write that point above all enzymes acts in duodenum of small intestine. That point you should remember. Okay, students, right. Now the digestion
Sir, by
process.
secreted which gland, sir?
Yes, students,
Sir, bicarbonates are secreted by which gland, sir?
bicarbonates by the epithelium of mucosa, secreted from the epithelium of the mucosa only. Clear, students?
Yes.
Right. So we came to know what are the list of pancreatic enzyme. Now the enzymatic reactions. The digestion, first digestion process of proteins, carbohydrates and lipids by the help of pancreatic juices, pancreatic enzymes. So first one, digestion of proteins. Digestion of proteins. Digestion. Okay. So we already discussed it in earlier sessions where the digestion of proteins are initiated.
Stomach.
In which part of the gut? The region of the stomach. The digestion of proteins are initiated, started in the region of stomach. So the proteins are converted into, yes, students, proteins are converted into in the stomach.
Proteoses,
Peptones and peptides.
Proteoses and
Stones and proteins.
proteoses and peptones. By the influence of which enzyme, students, in the stomach?
Pepsin.
Pepsin. So the remaining proteins and whatever the proteoses and the peptones need to be converted into amino acids. That process is takes place in the duodenum. So we already discussed that any type of the enzyme has gen in the last, pro in the first. This indicates what, students?
Inactiveness.
Inactive state.
So first one, the inactive enzyme. First, before going to the enzymatic reactions, inactive enzymes need to be converted into active
Active
form.
Active enzyme.
Right. So first in this protein digestion in duodenum, first inactive enzymes, trypsinogen, chymotrypsinogen, and procarboxypeptidase need to be converted into active form. Clear? Right. So first what happens? Trypsinogen converted into active trypsin by the influence of enterokinase enzyme. Enterokinase enzyme secreted from the glands of small intestine, from the crypts of Leberkuhn. We discussed already. So at the base of the villi, crypts of Leberkuhn is presented. Okay, students. So in the duodenum, the glands crypts of Lebercun. From the crypts of Lebercun, what secretes? Enterokinase secretes. Enterokinase is the enzyme of what? Enterokinase is the enzyme of not pancreas. It's secreted from the intestine. That point you need to remember. So inactive trypsinogen is converted into active trypsin by the presence of what, students? Enterokinase. Influence of enterokinase. In the pH is 7.8 pH is need to be maintained. Alkalinity 7.8 pH. Okay, students. So write down here enterokinase enzyme presented in intestinal juice. Intestinal juice secretes from crypts of Lebercun. Okay, students. So what are the intestinal enzymes? We will discuss later. So first, so what type of how the protein digestion is initiated? How the protein digestion is initiated in the region of duodenum? That is first one, trypsinogen. Inactive trypsinogen is converted into active trypsin in the presence of enterokinase. That point you need to be remember. So enterokinase secreted from pancreas or intestine?
Intestine.
Intestine only. Okay. For avoiding of confusion, you need to mention the point here. Okay.
Sir,
Right.
will I present it
Now-
in duration, sir?
Questions last 10 minutes, man. Last 10 minutes question. Let me complete first. Okay. No need to worry. If you want more time, I will give 15 minutes also. Right. I will clarify all of your questions. Right. Next one. First, trypsin activated. Once trypsin activated, the trypsin actives remaining two inactive enzymes of pancreas. What are remaining two inactive enzymes of pancreas, students?
Chymotrypsinogen, carboxylpeptidase.
Procarboxylpeptidase. Right. So write down the second reaction is the chymotrypsinogen. Chymotrypsinogen is converted into inactive chymotrypsinogen converted into chymotrypsin. Clear, students? In the presence of
Trypsin.
trypsin. Same pH. In one reaction, you need to mention the pH value. For all reactions, not required, because all the reactions, enzymatic reactions, are to take place in the region of duodenum only. So same maintenance of pH. Next one, inactive procarboxylpeptidase. Procarboxylpeptidase is converted into active carboxylpeptidase. Inactive procarboxylpeptidase is converted into active carboxylpeptidase by the influence of which enzyme, students? By the influence of same trypsin only. So what is the function of trypsin? What is the function of trypsin in the region of duodenum?
Digestion of protein and conversion.
To activate
It
the
converts the inactive enzymes into active enzymes.
Convert the inactive enzymes into active enzymes. What are they? Chymotrypsinogen and procarboxylpeptidase. That point is important. Okay, students. So these two are inactive forms and converted into active forms. So now what happens? Trypsin activated, chymotrypsin activated, and carboxylpeptidase is also going to be activated. By the influence of trypsin, chymotrypsin, and carboxylpeptidase, the protein digestion is initiates here. Till now, what we discussed, inactive enzymes are converted into active enzymes. So protein digestion is not yet initiated here. Right, students? So next reaction, the proteins and peptones and proteases. Where the peptones and proteases are formed, students?
Stomach.
So by the influence of enzymes of trypsin, enzymes of chymotrypsin, and enzyme of carboxypeptidase enzymes, all are converted into dipeptides. So in this reaction, fourth reaction, protein digestion is completed, students.
No, sir.
No, sir.
Has protein digestion completed? No. Why? Because what are the end products of the proteins?
Amino acids.
Amino acids.
The acids are formed here.
Dipeptides.
Dipeptides.
So what point you need to remember, students? The pancreatic enzymes which are involved in protein digestion is converts up to some of the stages only, intermediate stages only. So which enzymes are helpful for completion of protein digestion? The enzyme secreted from
Intestinal
Small
digestion.
the enzyme secretes from the small intestine. Are you understanding or not? Okay, students. So here the protein digestion in the region of the duodenum with the help of pancreas only now we discussed. So the pancreatic enzymes already we came to know. So first inactive enzymes are need to be converted into active forms. Inactive trypsinogen converted into active trypsin by the presence of enterokinase. In the pH value 7.8 pH. So enterokinase enzyme secreted from the intestinal glands, that is presented in intestinal juice. And once trypsin activated, so what is the function of trypsin? Both chymotrypsinogen and procarboxypeptidase is need to be converted into active chymotrypsin and active carboxypeptidase in the presence of trypsin. Okay, students. Next one. Once trypsin, chymotrypsin, and carboxypeptidase is formed actively, then proteins, peptones, and proteases are converted into dipeptides by the influence of students, trypsin, chymotrypsin, and carboxypeptidase. Until this clear, all of you? So this reactions are takes place in the region of duodenum of the intestine, small intestine, with the help of which juices, students? With the help of which enzyme, students? Intestinal enzymes or pancreatic enzymes? Pancreatic enzymes. That point you need to remember. Okay. So till now we discussed regarding about digestion of proteins in duodenum with the help of pancreatic enzymes. So second one, digestion of carbohydrates. Digestion of carbohydrates. So digestion of carbohydrates, here what happens? Is there any pancreatic enzyme is there for digestion of carbohydrates, students?
Pancreatic amylase.
Pancreatic amylase.
Amylase.
Pancreatic amylase is there, right, students? Okay. So here what happens? So what is the function of amylase? So first reaction, carbohydrates, or you can write here in the brackets, starch or polysaccharides. Carbohydrate, those are polysaccharides, are converted into disaccharides. In the presence of, yes, students, in the presence of which enzyme?
Yes.
Amylase. Which amylase? Pancreatic amylase. So now carbohydrate digestion completed or not, students?
No, sir.
Not
Not.
complete.
No, sir.
What are the end products of carbohydrates?
Glucose.
Glucose
Glucose.
or monosaccharides.
Not yet completed. So disaccharides only formed. Okay, students, right? So disaccharides are need to be converted into
Monosaccharides.
monosaccharides by the help of which enzymes?
Intestinal enzyme.
Intestinal. Very good. Until this clear, all of you, right? Next one. Next one. Digestion of fats. Digestion of fats or lipids. Before fat digestion, the fats are undergoes the process of emulsification by the help of bile juice secreted from the liver. So what is the function of bile juice? Already we discussed. The bile juice participated in the
Yeah,
process of
emulsification of fats.
emulsification of the fats. Okay. So what is first reaction is takes place? Bile, fats or lipids undergoes emulsification process in the presence of, yes students, in the presence of
Bile juice.
juice.
Yes.
What are formed? Emulsified fats are formed. Emulsified fats are formed. These are called mycelials. What is meant by mycelials? How the mycelials are formed? The fats are undergoes emulsification process by the influence of the bile juice. Emulsified fats are formed. Those are called mycelials. One more question, most important. So where the mycelials is formed, which part of intestine mycelials are formed? Yes, students? Which part of intestine? These are all takes place.
Duodenum. Duodenum.
Part of duodenum.
So for the mycelials, for the formation of the mycelials, which enzyme is helpful? For the formation of mycelials, which enzyme is helpful? Answer is none of this. Which enzyme is helpful? No enzyme is helpful. Only digestive juice is helpful. Bile juice is
Bile
not
juice.
an enzyme. Bile juice only. Clear, students, until this? Okay. What is meant by emulsification? Emulsification means
Breaking of large fat globules.
okay. Decreasing of surface tension of the fats. Suppose on the cloths any stains are there, what we apply? We'll apply detergent or not?
Yes.
When we apply soaps and detergent, stains will remove or not? The same process of emulsification is takes place by the bile juice. So emulsification means decreasing the surface tension of the fats. Whenever fat surface tension is decreases, then obviously easily fats are going to be digested and fats are involving in further steps of digestion process. Are you understanding or not? Okay, students, so here write down emulsification. Emulsification means decreasing of surface tension. Decreasing of surface tension of the fats are called emulsification process. So before fats digestion, what takes place? Emulsification processes takes place. Fats compulsory, emulsified fats only going to be participates in further digestion process, further enzymatic reactions. Otherwise, directly fats are not participated in the process of digestion. So that only in the stomach there is gastric lipase is formed, so that complete process of fats are not takes place because the stomach cannot receive what? Bile juice. Understanding or not? Yes, students?
Yes, sir.
Okay. Next, the second reaction. So emulsified fats here. Emulsified fats. So which enzyme helpful for fat digestion? Yes, students? Amylase or lipase?
Pancreatic amylase.
Very good. Pancreatic amylase.
Pancreatic, sir.
Pancreatic amylase.
Sir, lipase.
Sorry.
Lipase.
Pancreatic lipase.
Sorry, sir. Lipase.
Amylase. Pancreatic lipase. Okay, students. Pancreatic lipase. Emulsified fats by the influence of pancreatic lipase are converted into triglycerides. Triglycerides. So fat digestion is completed in the second reaction. Yes, students, completed or not?
No, sir.
Sir.
No, sir.
No, sir.
What are the end products of the fats?
Fatty
Fatty
acids.
acids and glycerol.
Glyceride.
Below fatty acids and glycerol are not formed. So some of the enzymes which are secreted from the intestine, which are need to be converted into triglycerides into, that is fatty acids and glycerol. So that by the secretions of the pancreas, some of the digestion process only occurs. That our final digestion not occurs. So final digestion is occurs by finally secreted in the region of duodenum by glands of duodenum. Okay, students, until this clear all of you, right? So once again, I am revising these points. Digestion of carbohydrates. Digestion of carbohydrates already initiates at where, yes, students, already initiated at
Buccal cavity.
right. So remaining the carbohydrates, the polysaccharides are converted into disaccharides by the presence of pancreatic amylase enzyme. Okay, students, right. Next one. So disaccharides are not the end products of carbohydrates. So these need to be converted into monosaccharides. Next one, digestion of the fats. Before fats are involved in the enzymatic reactions, fats are need to be undergoes the process of emulsification by the influence of which juice, students? By the influence of the bile juice. So fats by the influence of bile juice, emulsification process, emulsified fats are formed. Those are called mycelials. And next one, what is meant by emulsification? The decreasing of the surface tension of the fats is called emulsification process. Next one, emulsified fats are converted into triglycerides. That is by the influence of pancreatic lipase enzyme. Until this clear, students?
Clear, sir.
Sir, how the surface tension will be decreased, sir?
Questions last 10 minutes. No more explanation between the class. Next one, students. Carbohydrate, protein discussed, carbohydrate discussion, and fats also discussed. Next one. Which material remains, students? Take the list of pancreatic
Nucleic acids.
Nucleosis.
Yes.
Nucleic acid.
So, digestion of nucleic acids. Digestion of nucleic acids. Okay, so here what happens? The nucleic acids are converted into nucleotides. And next one, the nucleotides are converted into nucleosides. By which enzyme, students? Nucleases. Nucleases secreted from excuse me.
Yes.
Nucleases are secreted from the, yes, students? Pancreas. By the
Pancreas.
nucleases, deoxyribonuclease and oxyribonuclease, the nucleic acids are converted into nucleotides and nucleoside by the presence of nucleases enzymes. Until this clear, students? So these are all reactions, enzymatic reactions, which are takes place in the region of the duodenum by the influence of pancreatic enzymes and by the influence of the bile juice. Okay, students, until this clear? And before going that, one more point we need to be discussed here regarding about bile juice. We need to discuss what are the components are presented in the bile juice. We already discussed that bile juice do not have any type of the digestive enzymes. So what type of the components are presented in the bile juice? So bile juice secretes from where, students?
Liver.
Liver.
So bile juice consists with bile salts and bile pigments and cholesterol and phospholipids. So what are the components of the bile juice? Like a bile juice, what are the components? Bile salts are presented, bile pigments are presented, cholesterol is presented, and phospholipids are also presented. So bile salts, what are bile salts? Sodium tarcholate, and sodium glycolate. So what are the names of the bile salts? Sodium tarcholate and sodium glycolates are the bile salts. Next one, bile pigments. The bile pigments are, yes friends, anyone? Bilirubin
Bilirubin and biliverdin.
and biliverdin. Bilirubin and biliverdin, those are bile pigments. So bile salts, sodium tarcholate and sodium glycolate, both are helpful for the process of emulsification. So how surface tension is decreased? Because of the salts are presented. Which salts? Bile salts are presented. Sodium tarcholate and glycolate. So that only the surface tension of the fats are going to be decreased by the process of emulsification. Next one, bilirubin and biliverdin. So bilirubin and biliverdin, two functions, participates in the process of color 2, participates in the process of formation of RBC, Second one, providing color to fecal matter, fecal material. By the presence of bile pigments only, fecal material has some of the distinct color. Otherwise, in absence of bile pigments, the fecal material turns into the color of white color. Clear, students? So here, bilirubin and biliverdin provides participation of the process of formation of the RBC. Yes, students. So production of hemopoiesis, erythropoiesis, and next one, the bile pigments providing color to the fecal matter. So those are the functions of bilirubin and biliverdin, bile pigments, and as well as bile salts. Until this clear, students? Yes.
Yes, sir.
Okay.
Yes, sir.
What are the process of remaining the intestinal enzymes? So what are the enzymes secretes from the intestine? And the list of enzymes and functions of the enzymes, the enzymatic reactions which are takes place in the region of duodenum, we will discuss in next session. Until this clear? Yes, you can ask any doubts.
Sir,
Yes,
will I?
So
Duodenum.
formation of RBC takes place in bone marrow, no, sir?
Bone marrow. But these are helps. It goes to the liver, presented in liver. Liver is also one of the major organ which are helpful for formation of different types of the blood cells. The WBC or RBC platelets, liver also participates. Process is takes place in the bone marrow.
Sir, in buccal cavity only.
But at the time of the embryonic stage, the complete blood cells production from liver only. After birth only, the production of blood cells from bone marrow. Yes, students.
Sir, in
Sir,
buccal
will
cavity only the
I-
polysaccharides are converted into disaccharides, no sir?
Yes. How much percent?
Sir, how it is reversed, sir? Again polysaccharides are converted into disaccharides.
30% only happens.
30% only, no,
30%
sir.
only, and amylase function is that much only. Amylase function is what? Amylase is able to convert the polysaccharides into disaccharides. That's it. That disaccharide to monosaccharide, that power is not there for amylase.
Sir, the remaining 70% is digested now, sir.
Yes. Remaining 70% digested.
Sir, will I
That is also important.
sir?
Remaining 70% final digestion by intestinal enzymes. That point you should remember. So that only up to pancreas I had concluded. You understand or not?
Yes, sir. Yes, sir.
Sir, duodenum lacks villi, sir.
Duodenum?
Lacks.
Duodenum are not there. Only glands.
So
Villi?
then how the crypts of liver can secrete sir, enzymes, sir?
Villi is not there. Villi is for what? Villi is for only
Absorption.
absorption. When absorption takes place after the completion of digestion. Where completion of digestion is there? Duodenum. So, in jejunum, more villi are there. In ileum also, less villi are presented. So, jejunum and ileum, crypts of liver are absent. Are you understanding or not?
Okay, sir, in only duodenum crypts of Leberkan are more.
Yes. Complete digestion secretion means duodenum only. In the region of jejunum and ileum, villi for absorption.
Sir,
Okay.
sir, you told that villi will be not present in
What
duodenum.
about-
Sir.
Villi?
Will not present in duodenum part, no, sir.
Yes.
Sir, then how that enterokinin will enter into duodenum, sir?
Enterokinase?
Enter into duodenum, sir.
Enterokinase enter into duodenum by secreted from glands, duodenum glands only. It's not secreted from villi. Brushwater epithelium of duodenum, that's it. See, villi is different. Villi is for only absorption. And crypts of liver for what? Only secretion of enzyme. This is different, that is different.
So what is the necessary of digestion of nucleic acids, sir?
If nucleic acids are not converted, nucleic acids are formed by what? Pentose sugars, nitrogen bases. Not proteins. Pentose sugars and nitrogen bases.
Pentose sugar base.
Pentose sugar, nitrogen bases are the end products. If nucleic acids are not converted into pentose sugars and nitrogen bases, the process of digestion is not there. Still, those are macromolecules only. DNA, RNA, macromolecules that need to be converted into absorbable forms. So that only required.
What are the absorbable forms, sir?
Those are only absorbable forms. Pentose sugar and nitrogen bases.
Okay, sir. Thank you, sir.
Okay. Next one.
Sir, what about the decreasing of surface tension, sir, in fats? How it will decrease, sir? Emulsification.
Emulsification, how it will decrease means what? What happens? Fats, the chemical bonds are more. Triglycerides are converted into triglycerides are converted into monoglycerides. So there is more chemical bonds are there. Whenever breakdown of the fats, the surface tension is need to be decreases. Surface tension means what? Whenever emulsification, after the emulsification only, the fats are involved in enzymatic reactions. In case emulsification process not occurs, then what happens, students? Fats are not involved in enzymatic reactions. Fats are not involved in enzymatic reactions, different enzymatic reactions. It's not possible because of surface tension is more.
Sir, pancreatic
But
amylase.
to break the bond,
Sir,
to break the attraction, the emulsification takes place, sir.
It is ready to be process of enzymatic reactions. Emulsification means.
sir, at what pH pancreatic amylase will activate, sir?
Same.
Okay, sir.
Pancreatic amylase means same pH. These are all reactions.
6.8 or 7.8, sir.
That is 7.8 only. Alkalinity.
Yes, sir. Thank you, sir.
Either bile juice, apart from the stomach, remaining all our alkalinity is more. 6.8 to 7.8 more alkalinity. There's no acidity. Acidity in the region of
Stomach.
stomach only.
Sir, what is the purpose of cholesterol in the bile juice, sir, in our body? What does it do, sir? Cholesterol.
Cholesterol is different types. There's good cholesterol, bad cholesterol is there. LDL, HDL. So cholesterol is required.
Why is it necessary, sir?
Cholesterol is storage energy, one of the storage energy.
Okay, sir. Thank you, sir.
Right. Next
Thank-
student. That's it.
Sir, function of phospholipid is the breakdown of lipids, sir.
Breakdown of the lipids. Yes. Yes, students.
Sir, crypts of lubricant, they produce enterotinase, sir.
By the mucosal glands. Yes. Mucosal glands of duodenum.
Duodenum.
Yes,
sir. No villi is present, but only the crypts of lubricant are present.
And one more important, mucosal glands produces intestinal juices and submucosal Burners gland in duodenum. The two modifications you need to remember.
The Burners glands also produce mucus, no, sir?
Mucus, we discussed already. Burners gland secretions helps for what? Maintenance of alkalinity.
Alkalinity.
Alkalinity. That only, alkalinity only, there is enzymatic reactions are takes place.
Sir, can we say mucosal layer is modified into intestinal glands, sir?
Mucosal layers are modified into intestinal glands, and submucosal layers are modified into which intestinal glands means Burners glands. Remaining all are intestinal
Runner
glands.
gland.
Yes. Modifications important, secretions important.
Okay, sir.
Okay, students, this weekend you don't have any examinations because of due to the eve of what? Dasara?
Sir, Diwali.
Diwali. Okay. So exclusion given from our college. Okay. So exam is not there, but reading compulsory. You should read on daily basis. Otherwise, you will forget for tomorrow only. Immediately, you should revise all the points of NCERT and running notes. Okay, student, that is most important. If exam is not there, no need of opening a book means no use. Okay, students?
Okay, sir. Thank you, sir.
Okay, students.
Bye, sir.
Advance happy Diwali, sir.
Bye, sir.
Thank you, sir. Bye, sir.